second one we've had this year. So Joe was a speaker in our first BCBT summer school, and he does fantastic work on underwater robots. And like myself, I think his main motivation is to use robots to explore biology, and he has a really quite distinct approach of modeling complete neural circuits for these underwater animals that he's looking at. And he built some really remarkable robots by doing this. So over to you, Joe.
Okay. Well, what I'm going to talk about today is going to draw on stuff I've done in the past, but I've recently gotten some new funding, which is just starting, which really focuses on an intersection between neuroscience, robotics, and synthetic biology. So that's why I chose the title Synthetic Neuroethology. And I'll give you some idea about what synthetic biology is. It's a kind of a new discipline, and I think it's going to really revolutionize the way we can make robots. Now, my background is actually, I'm pretty much a straight neurophysiologist, but I come from an area of neuroscience called neuroethology. And what we try to do is to explain the behavior of simple model animals in terms of the properties and connectivity of networks of neurons. And we're especially interested in how they adapt to contingencies of environment. And recently we've been focusing on how to build devices that capture the performance advantages of the animal models in natural environments. We also eat the experiments. Now, the models we choose have very different nervous systems than vertebrate nervous systems. Their nervous system is quite distributed, and they have a segmental metamerized body plan, and they have separate little mini brains or ganglia that control each of the body segments. And that's an incredible technical advantage. Another advantage of these animals is that there's no evidence at all that they have a pain afferent system. So you can cut them up into pieces, and the pieces often operate quite effectively by themselves, either in vitro or in situ. So they really give you an access to the nervous system that you wouldn't otherwise get. We used to call them simple systems, and we no longer do that. We call them technically accessible systems. Now, this is an example of one ganglion here called the stomatogastric ganglion. And this is probably the best understood piece of neural tissue in the animal kingdom. Each of these round things you see here is the cell body of an individual neuron, and there's only 30 neurons in this ganglion. And the ganglion is covered with a sheath, like a pillow has a pillowcase, and we have to go in surgically and remove that sheath. And this distance here is 600 microns. So for scale, a human round head hair is about 110 microns. So these are tiny things. And this photograph is taken at 80 diameters magnification. So it's a pretty small little mini brain. Now, the real advantage of these invertebrates is that by a variety of criteria, you can identify each neuron. And we can do this by the projections that the neurons make out to muscles. We can do it by the unique waveforms that we see in the neurons, and also by their patterns of connectivity. But every ganglion in this, or every neuron in this ganglion has a name, and we can study it not only from animal to animal of the same species, we can study it between species of crustacea. When you have identified neurons, you can record from pairs of them and trigger on a presynaptic action potential and see the postsynaptic spike. So these are two neurons that are reciprocally inhibitory. An action potential in this neuron causes an IPSP in this one. An action potential in this neuron causes an IPSP in this neuron. If you do this with all possible pairs of two neurons, now think about this. This is a ganglion with 30 neurons. This is the network that you see among 30 neurons. So each one of these objects represents an individual neuron, and these are all the connections. I'm always fascinated by people that tell me they're going to explain how the cortex works. And this is why we no longer call these simple systems. But they do give us incredible technical access. We can record from up to 8 neurons intracellularly at once. We can do perturbation experiments where we apply stochastic stimuli at different phases in the cycle and look at phase response curves, for example. But most importantly, this ganglion is under control by external neuromodulators, and there are neuropeptides. Here's an example of the effect of one called proctolin, and it not only alters the properties of individual neurons in the network, it also alters the synaptic strength of particular synapses within the network, and different neuromodulators produce very different behavioral patterns. So they change the context of operation of the nervous system so that the same piece of neural tissue can subserve a variety of behavioral acts. Now, what came out of about 40 years of study of these is a model called the command neuron coordinating neuron central pattern generator model. And I was lucky enough to be a PhD student of the guy who did the command neuron work and then be a postdoc with the guy that did the central pattern generator work that established this model. And this argues that the controlled plant is controlled by a central network of neurons that consists of motor neuron pools that activate individual muscles, typically a neuronal oscillator, and in some cases what we call a pattern generator, which can reconfigure the network so it produces different patterns of output. In multi-segmental animals, there's a set of central pattern generators for each body segment, and these are coordinated among themselves by a class of neurons called coordinating neurons, which apply information from a governing oscillator to a governed oscillator to alter its temporal output so they remain in a pattern of coordination. This pattern of coordination is typically to maintain a pattern of constant support against gravity. So for example, insects will walk in what we call an alternating tripod gait, so they're stable both in the pitch and the roll plane. Then there's a third category here of what are called command neurons. And these are neurons that are typically in the brain. They're cell bodies in dendritic fields. And they're the point at which the decision to evoke a behavior is made. And they descend down into the thoracic chain and turn on the central pattern generators to evoke a behavior. Now, these were first described in 1939 by a guy named Kees Wiersma. And the giant fibers that cause the tail flip escape of a crayfish was the first identified neuron in the animal kingdom. Now, I want to show you some examples. Oh, the other point I want to make is that this is a general model. It applies to all bilaterally symmetrical animals. We can take these same components and reconfigure them to form a central pattern generator for an undulatory organism, the sea lamprey. If we took and say for every alternate pair, put a walking CPG, we could produce a central pattern generator for an alligator. So this is a conserved model that applies to all animals, and in fact these components provide sort of an erector set for building any kind of animal-like controller. Now I want to show you some command neurons in action. Now this is an experiment where the brain connectives have been exposed and they've been frayed into single fibers like you'd fray a rope into fibers. If you're lucky, you can pull one of these fibers up onto an electrode, and when this light comes on, it's stimulating a single neuron that is here a postural command neuron. It causes the abdomen to extend, and there's a family of about 20 of these, each of which makes the abdomen go into a different posture. So they quantize their behavioral output. They have a series of different states that they switch between by switching to different neurons. Well, there's a little crosstalk between them, but in general, they produce one pretty unique behavior. Okay, and that's the other stuff is kind of in the noise level. It's present from simulating pretty much all of these. So this is a forward walking command that also turns on swimmerette beating. And then here's another command neuron that causes the swimming escape response, multiple tail flips. So the activity evoked by these neurons represents the least divisible unit of behavior. So it's our task to try to string together these sort of atoms of behavior to make molecules, which are what the animal does in the natural environment. Now we do a process we call biomimesis, where we try to go from the animal model to a biomimetic robot. And this movie here is the first time we ever turned this robot on. So this was actually the second generation of our lobster robot, but we pretty much got it right at this point. And I love to show this movie because it's very rare when something works when you build it, I'll tell you that. Now these are really quite unique. They take the form of the animal because the form of the animal gives them biomechanical advantages for dealing with flow, for being able to walk in all directions. We use a neural circuit-based controller. We use neuromorphic sensors that code with a labeled line code. We use artificial muscle that in our case is a shape memory alloy. And then we create a library of behaviors that's based on reverse engineering the behavior of the model animal. Now the other animal model we use is a sea lamprey, and this is a very primitive vertebrate. It's in between Amphioxus and elasmobranchs. And the real advantage of the sea lamprey is there's no myelin in the nervous system. So the nervous system is completely transparent and you can see all the neurons in the spinal cord like you can in an invertebrate. So this gives us quite the same technical advantage, but it really has a brain. It has a thalamus. I don't know if it has a superior colliculus, but it has most of these other brain structures that you see in higher vertebrates. It does. Yeah, I know it has cerebellum. Yeah. So this is our lamprey-based robot that we built. This was in about 2000. This is the first generation. And at this point in this research, I became aware of a real conundrum. If you try to control robots algorithmically with computer programs, unless you anticipate every possible situation the robot is going to get into and have programmed a detailed escape strategy, it's going to get stuck. And I can guarantee you this happens because it's happened a lot to us. Animals never get stuck. If animals got stuck, the trees would be full of birds, the bushes would be full of cats and dogs. It just doesn't happen, okay? Now what animals can do that current robots can't do is they can wiggle and squirm. And we think that what the animals are doing, in fact we have very good evidence for this, is that they're increasing the level of chaos in the nervous system, and the chaotic variations on the locomotory programs are the wiggling and squirming. So if we can build robots that can wiggle and squirm, we can build robots that aren't going to get stuck. So to get a handle on this, I went out to a place called the Institute for Nonlinear Science. And the Institute for Nonlinear Science consisted of a bunch of physicists. The leader was a guy named Henry R. Bobanel, and he's one of the Jasons. He's one of the 30 physicists that control the United States nuclear deterrent. And he got together with a guy named Misha Rabinovich, who was the head of the Laboratory of Radiophysics at Nizhny Novgorod, was the chairman of the Soviet Academy of Sciences, and was on the Vatican Scientific Advisory Council representing the governments of the United States and Russia. I mean, these guys are real heavies. And they got together with a couple of biologists, which are Yuri Arshevsky, who used to run the Laboratory of Neurophysiology at Moscow State University, and my old postdoctoral mentor, Al Silverstein. And they started trying to apply nonlinear dynamical models to the nervous system. So the first question that the physicists asked the biologist was, how many degrees of freedom does a neuron have? Now, neurons have ionic channels, and the channels have gates, and they have gating currents and all sorts of biophysics. And a really interesting neuron will often have 6 or 8 different families of ion channels. So I would have said, hey, it probably has 150 degrees of freedom. Okay, now if you try to model that with differential equations, which is what the physicists wanted to do, it's going to be really complicated, and you're not going to be able to model very many neurons at once. So they got Al Silverstein to isolate some neurons from all sources of synaptic input, but keep the modulatory inputs active. And he did this by taking all the known presynaptic cells and injecting them with a dye and shining intense blue light on them, which makes them die. And when he isolated these cells from synaptic input, they went into truly chaotic patterns of activity. So what they observed was that if they fooled around with an internal calcium buffering system called the inosine triphosphate system, which is a pump that's resident in the endoplasmic reticulum, they could switch them from chaotic activity to regular bursting. And when they did that, they did a form of analysis called local faults nearest neighbor analysis, and they were able to demonstrate that in the regular bursting case, they had only 3 degrees of dynamical freedom, and in the chaotic case, they had 4 degrees of dynamical freedom. So they came up with a set of equations that were first based on some equations that came from a guy named Rose that worked with Paul Benjamin and an astrophysicist named Hindmarsh, which were called the Hindmarsh-Rose equations. And these describe the dynamics of the membrane potential, the fast membrane events of the action potentials, the slow membrane events of the slow waves, and then finally the UCSD guys added this term for the slow calcium dynamics. And they instantiated this as an analog computer with 4 integrators. And each of these circles represents X, Y, Z, and W. And then they built physical boards that realize these analog computers. And when you plug them into an oscilloscope, they're largely indistinguishable from a living neuron. Now, the interesting thing here is that one of these trim pots here, which controls some of the internal voltages, varies a term called E. And when you vary E, you can switch the neurons from regular bursting to chaotic activity, which fulfills this requirement that we wanted of being able to vary chaos. So chaos is now something that we can control with an internal voltage. So I took a model that I developed when I was in graduate school almost 40 years ago and instantiated that using electronic neurons and synapses. So these are boards that model the synaptic connections between the neurons. And sure enough, if you do that with 8 neurons, we can get them to generate the motor patterns. And I should go back here. Let me show you one thing. In this network model, there is a central pattern generator. These are motor neurons. These particular central pattern generator neurons are also motor neurons. And then there are 2 command neurons that gate off by presynaptic inhibition some particular connections in the circuit. So if you do that and turn on the forward command, you get a forward walking pattern. And if you turn on the backward command, you get a backwards walking pattern. So you see that these motor neurons are changing phase relative to this pacemaker between the 2 cases. And this is exactly what occurs during forwards and backwards walking. And if we take these neurons now and use an actuator, which is called nitinol. This is what's called a shape memory alloy, and this is a muscle. So a nitinol is a 50-50 alloy of nickel and titanium, and it exhibits a phenomenon called the shape memory effect, so this is temperature, this is the length of the muscle, and this is temperature. If you heat the muscle up to a very high temperature, it goes into a very dense crystalline state called austenite, and you can cool it down and then mechanically deform it by the action of an opposing muscle to where it undergoes about a 5% length increase. Then if you heat it up again, it will contract, and a 250 micron wire will lift a kilogram in about 150 milliseconds. So it's an incredibly force-dense material. Now, normally when you operate in this area, you have this problem of cooling it, which is a nightmare. But if you're operating it underwater, you can get it to cool down in about 200 milliseconds. So we can get these things to operate up to about 3 hertz underwater. So they have really good response dynamics. And if you vary the amount of current by pulse width duty cycle modulation, this is the upward displacement of a 1 kilogram weight. You can both vary the contraction velocity as well as the amplitude of contraction. And if you make the weight go above what the wire can bear, it'll quickly switch from an isotonic contraction to an isometric contraction. For all intents and purposes, underwater, this is just like real muscle. But the way we achieve excitation-contraction coupling is we take the action potentials and we threshold them with a comparator circuit, and then we generate a square wave pulse which activates a power FET, and that applies current to the nitinol actuator. So here we use heat in the same way that muscle uses calcium to achieve excitation-contraction coupling. And if you look at the way most artificial muscles are controlled, the weak point is always excitation-contraction coupling. That's the hardest thing to achieve. Now this is one of the legs of my robot now being controlled by that 8-neuron circuit to walk forwards and backwards. Now if we added 4 more neurons, we could also make it walk sideways with a leading side and a trailing side. So this really produces a lot of behavior with a very, very, very simple circuit. Now, the problem with those electronic neurons is that, yeah.
You have a defined limit for the movement of the arm,
Uh-huh.
which is just to make sure they don't hit each other, because I noticed that the legs, when they move.
That was the first time we ran the robot. When we tuned up the parameters a little bit, we got it perfect. But I'm so proud of that movie because it really worked out of the box, you know, which was, to have that happen is kind of scary. Now the problem with these discrete time map-based neurons is that they're physically large. I mean the Hyde-Marsh-Rose neurons, they use high voltages and you can't get very many of them in a robot. So one of my other colleagues, a true Siberian, Nikolai Rulkov, came up with an alternative model which is called a map-based model. And here you have some equations that describe a one-dimensional map. This is the membrane voltage in cycle n plotted versus the membrane voltage in cycle n+1. And it has a fast component where it loops around here to produce action potentials. And every once in a while, it slips down below from one nullcline to another nullcline, which generates the slow wave. And he has some fuzzy logic over different ranges of x. And if we vary two control parameters, which are called alpha and sigma, over the space of sigma and alpha, if we configure it in this region, the neurons are silent. If we configure it in this region, they generate bursts of spikes at constant frequency, and these points correspond to these traces here. In this area, it generates, so here's constant frequency, here it generates bursts of spikes of different period, and in this yellow area, it generates truly chaotic activity. This is the space that this thing oscillates around. So as you increment through n, that's what we call it discrete time, it's in a loop, and you recalculate where it is on this loop for every loop cycle. So for most of the time, it's running around this part, but given the effect of y, it drops down below this nullcline.
When you say this part here, you're going from the solid line to dotted line.
Yeah, it's just looping around here.
The dotted line is it's like a cobweb plot, so the dotted line shows the trajectory it will be taking.
Yeah.
So
exactly.
you can kind of ignore the dotted line, it just sort of shows the path.
It
just shows the attractors.
Yeah, it's very strict. You know, this is really interdisciplinary research. This is not something I would have come up with on my own, I guarantee you. and Nikolai is really quite a brilliant guy. He's a real chaos master, to say the least.
Is the underlying dynamics related to Isakovich neurons? Because I mean, they oscillate around phase planes and they split up the dynamical structure of the phase space.
So what is the question?
Is the underlying dynamics related to Isakovich neurons? Because that has a...
He actually came up with this before Isakovich did.
Okay.
And Isakovich is kind of an entrepreneur, if you know what I mean. Now, they also have some math for synaptic inputs, which computes this term beta, which is the input on these neurons from other neurons. And so the thing about this model that is quite unique is it's based on difference equations and not differential equations. So we can do about 2,000 neurons and synapses on one TI-DSP chip. So it enables us to actually construct models with large numbers of neurons. And one of the robots I'm going to show you has 400 neurons in it. So we can get pretty elaborate models. So this is indeed that robot. This is the Lamprey robot. And it consists of a watertight hull where we have the electronics, a strip of polyurethane which forms a body axis, and this is divided into segments, and there are nitinol actuators at each segment which are in a chevron arrangement that cause it to flex. And to get the necessary amplitude, we tie these down to the central line at 2 points to shorten the chords, and that gives us large amplitude bends for very small contractions. And then these elements here are buoyancy elements that are made out of syntactic foam. So this thing is statically stable in the water. If you shut it off, it just hangs there at normal pitch and roll. Then we have a hinge between the body axis and the hull that enables us to either lift up the hull or depress it, which creates a low pressure area that causes the thing to dive or climb. And we vary turns by regulating independently the amplitude on the two sides. And this is the entire electronics package. So there's a DSP chip, this is a sensor board, and then these are the low voltage and high voltage electronics that control the actuators. This is a battery package. And then this is a sonar stack that is used for a short baseline sonar array that allows us to home on a sonar beacon. So we wire up central pattern generators. This is a central pattern generator for one body segment, and they're connected by anterior and posterior projecting coordinating neuron connections. And if you turn this on and bias the excitation towards the anterior segments, you get a front to rear propagating wave. If you bias the excitation of the commands towards the rear, you get a rear to front propagating wave. So this becomes highly maneuverable with really quite a small number of neurons. We found that with 5 body segments, we could achieve what the behaving animal does with 100 body segments. Now, this is this thing swimming through the water, and it illustrates this short baseline array, and this is one of the main sensors. So the research on this was paid for by DARPA, and they're interested in mine hunting robots. So this short baseline array does a calculation that we call the maximum likelihood sonar estimation. This dotted line represents the midline axis of the hull, and this red vector represents the position of a sonar source. And this calculates using 3 hydrophones the deviation in azimuth and inclination of the source from the hull. So this gives us some capability now. And we take that analog signal and we do lateral inhibition to achieve range fractionation. So we have different neurons that code for different degrees of deviation. So we can have some fancy logic for large turns as opposed to small turns. And we end up with a set of 4 interneurons, or a family of 4, that represent the port and starboard azimuth and the dorsal and ventral inclination of the target relative to the hull. So given these sorts of sensors, we can start to wire together a brain. Oh, wait a minute. The other issue we have to deal with is impediment. So normally if the animal takes off in a swim, it would have a pattern of acceleration, and if they were impeded, they would not accelerate with the same pattern, and that error, the difference between those two is an error signal that tells us it's impeded at start. If it's swimming along and it swims into some seaweed, it's going to show a slow deceleration. And if it's swimming along and it collides with a rock, it's going to show a very rapid deceleration. So just by using a cheap accelerometer, we can determine those 3 different situations and come up with a set of interneurons that code those 3 types of impediment. Now, we can start to use these sensors to wire together a simple brain. And for our purposes, we have some descending commands that go down through all the body segments that recruit the amplitude of discharge of the flexors on that side. There's a fast forward command, a slow forward command, and a backward command on either side. And then we take some interneurons that respond to sensor input, and we map those onto those commands. So these might be ones that cause it to yaw to the port or rotate to port. And then we have various sensors that map onto those. So this is the input from a compass, and I'll talk about how the compass works when I talk about the robo-bees. And then we also have, these are bilaterally symmetrical. We have the inputs from the short baseline array of the azimuthal deviation, and then we can map on the impediment compensation neurons. And this is pretty much all the interneuron circuitry that you need to mediate homing on a sonar beacon in the face of impediment. So it's pretty simple, which sort of blew me away. And we're right now in the process of tuning this thing up and getting it annealed so it behaves appropriately for the Navy's purposes. Now the beauty of this thing is it starts with a $29 central processor, so it's really quite cheap. And we run these discrete time map-based models on this processor. But we don't program algorithmically. We just simply wire together the wire list and the dynamic properties of the individual neurons, and the behavior emerges from that. So it's really a network-based controller. And then we can use a serial to analog interface to drive the neuromuscular interface and analog to serial interfaces from all the input sensors. And we end up with really quite cheap robots. And to be honest, the most expensive part of these robots outside of the intellectual capital is the batteries. And the batteries for these things cost about 120 bucks. So the cost of building these, if you manufacture them in quantity, very quickly approaches the cost of the materials. So these are really cheap robots. Now, what's significant about that? Well, if you look at what's going on in the Department of Defense, one of my sponsors is the head of the Joint Robotics Command for the Pentagon. So he's above all the forces. And he told me that right now there are 8,000 autonomous system programs in the Department of Defense. Okay? Now these range from things like the Remus to Predators to helicopters to boats. These are all very large and expensive systems, and they're all controlled algorithmically. Okay? Well, they just completed a 4-year study of all of these systems, and they've concluded that algorithmic control is not working to produce autonomy. Now, one of the problems is that these things are so expensive, and this gives you an example of the kind of issue that they're dealing with. They put 13 of these Remus vehicles that cost about $500,000 each out on a mission, and they lost 4 of them. Now, they were able to recover 1 of the 4, and they did that with a marine mammal, which is what these things were supposed to replace. Now, the state of the art, as far as Cliff Hudson tells me, is to be able to recover your teleoperations link if you lose it. That's the state of the art for the Department of Defense's autonomy. Now the Predator, for example, when they're flying a Predator, there are 30 people operating it. There are 3 shifts of 10 people to fly that one airplane. It takes 1 person to fly an airplane. It takes 30 people to fly a Predator. So these become what they call inverse force multipliers, and they want force multipliers. And the bottom line is that all the effort, because they have this risk management issue, is put on teleoperation, and nobody is really studying autonomy with these vehicles at all. So to study autonomy, we really need cheap vehicles. And this is the latest version of our Lobster robot, which was designed to minimize the parts count and maximize the assembly capability of it. And this is this thing now operating, the leg system operating now with a network of electronic neurons. So we can do this just like we do in the lamprey. And in fact, we're applying a lot of the same technology in the lamprey to building these things. Okay, now how would you use these things? I mean, this is always the question I get. What would you do with these things? Well, what they're interested in is dealing with agents of harm. Now, the agents of harm could be a lost nuclear weapon. They could be all sorts of things. They could be mines, whatever. But they want a way of going after agents of harm where they can remove the human operator from the situation. So this would be a boat that we call the mothership that would have a well in the middle of it and a gantry crane and a separate robot we call the grasper. And the grasper would have a sonar beacon that would allow the robots to home on it so they could be recovered. And then we would put a bunch of marsupials in this that would consist of robo-lobster, robo-lamprey, and a new robot we're working on we call Robolone that's based on the abalone. And this is a static sensing and communications node, okay? And the idea is that these would all communicate with each other with sonar, and the Robolone would form a long baseline navigational array, so you could position these things, and then you could send up a GPS antenna, and these could actually tell you exactly where the robot is for a remote operator anywhere around it. And in operation, the way these would work is using the same system that bees use. So the idea is that we would set up an array of these Robolone that would constitute the long baseline array. We would send the vehicle on a search vector, which is like what the bee gets from the waggle dance, a compass heading and a duration to navigate on that heading. We would also give it a propensity that when we think it's where the agents of harm are, it would investigate everything it encounters. And if it's some distance away and is just moving in, it would just negotiate everything it encounters. So once it's completed a search vector, it would enunciate its position, it would be sent a new vector, and would go off on a random search. And by that way, you could have one operator controlling a bunch of these things at once. So you don't have to have these 30 operators doing it, and you can keep humans completely out of the loop. Okay, we'd also like to get these things to do group behavior, and there are numerous examples of group behavior in the animal kingdom. This is queuing in spiny lobsters. I always like to show this to the military guys. They love this image. But the most interesting one is stigmergy. And stigmergy is, these are some casts of the nests of fire ants of different ages. And what you can see here is that this fire ant thing has a helical gallery in every case and these funny little side chambers. Now what's interesting about this is that there is no architect and there's no plan. Okay? So this thing has to emerge from low-level interactions of the individuals and affordances of the structure. And these interactions have to take the form of exteroceptive reflexes, which is the basis of our control architecture. I mean, everything we do with our robots is based on exteroceptive reflexes. So if you want to get at stigmergy, you need to deal off the top with social insects. So we have this other project, which is a $10 million NSF Computer Expeditions grant to basically build robotic bees for pollinating crops. So this consists of a bunch of engineers from Harvard, Jeff Barrows, who builds optical flow sensors, and myself. And we have a bunch of components that we're assembling into a robotic bee. And these consist of optical flow sensors, ocelli, the control actuators, the power electronics, the power source, the airfoils, and then the brain. And this is this thing actually taking off. So you can see it can fly. It needs a little control, as you might imagine, and this is just what we're getting into right now. And this is the monolithic airframe, and this is actually built in one procedure. They build these things in sheets and they self-assemble, and I can't tell you how they do this because they're in the process of publishing it, but this will give you the idea, the level of detail that they... What?
What make it?
It's a piezoelectric actuator right here. Okay, so I'm on, this team is divided into a body team, a brain team, and a colony team. So I'm on the brain team, and this is the basic circuitry that we're going to use for the brain. And at scale, it will fit right on the system right here. So we can produce a pretty small brain, a very low power brain, so that the power of computation is much, much, much lower than the power of actuation. And this is the basic accelerator architecture that we're going to use to control this. It has a Cortex M0, which is where the colony team lives, and then the brain part or team that does the actual flight control. We live here, and we're actually working on now a compiler to compile discrete time map neurons for this accelerator architecture. And we're hoping to use organizational rules from development so we can actually get this thing to self-organize. Yeah?
One simple question. Is this using low voltage VLSI as control?
Yeah, very low voltage VLSI. Yeah, these guys all come from distributed sensor network world, and they are really focused on the low voltage part. Okay, now what this thing is supposed to do during actual operation are what we call behavioral sequences. And the first one is flying away from the hive on a search vector and locating flies using UV photodetectors. So during daylight, flowers under UV light are very brightly colored and the rest of the world is gray. So the flowers really stand out from the rest of the world. Then they're just going to kind of bump around on the flowers to try to get some pollen. When they've done that, they're going to return to the hive using the inverse vector, and we're going to have a flashing LED on the hive that's tuned to the same UV wavelength as the flowers are. And then they will dock and feed, get a new search vector, and go out on a mission again. So the rules of the optomotor control of flight have been worked out very well by Srinivasan. And basically, he argues that they negotiate narrow passages by balancing the optical flow of input from the flanking walls. They control the speed by regulating the image velocity, and this enables them to flow through gaps. They land by keeping velocity constant as they approach a surface, and then they use visual odometry to gauge the distance. So these are the things that we need to embody in our electronic nervous system. So the way we're dealing with optical flow reflexes is actually taking a network that I also worked on when I was a graduate student 40 years ago. And the idea is that we take optical flow interneurons, and these were first described by Wiersma in the 60s, and they're now implemented by Jeff Barrows with his optical flow sensors. And then these are mapped on flight commands for the two sides. So they show a decussating connection, and these mediate height and velocity regulation. And then we have a second high velocity set that turn the first set off, and they mediate avoidance reflexes to get around obstacles in the environment. So if they start to get close to something, the optical flow velocity is going to go way up. That's going to switch to this, which is going to switch to an avoidance reflex as opposed to a approach reflex. Now, for heading interneurons, normally the bees communicate this using the waggle dance, where they dance at a particular orientation relative to vertical, and that's mapped onto the sun compass direction away from the hive. And we have a very simple linear approximation that we can use to calculate the amount of amplitude we need to apply to an interneuron based on a desired and an actual heading. And that gives us, enables us to code for a lateral bias. So we have ones that code for a heading deviation to port and one to starboard. And then we range fractionate that so we can have different levels of logic depending on how fast or how great the deviation is. So we ignore these sorts of deviations, we yaw here, and we reverse course when we're in this area. Okay, now odometry is something that we're just getting into right now, and this to me is really the sticky wicket. This is one of the hardest things to deal with. Dealing with time and neurons is not easy. So we have two approaches. One is a leaky integrator approach, where we have a odometry neuron that fires whenever it's gone the proper distance. And the other one is sort of cheating, where we just count spikes. Okay, so as you're flying out, you go for a certain number of spikes, then you switch to actually searching for the flower, and then you return on the inverse vector for the same number of spikes. Now, what I thought would be the hardest thing was controlling motors with neurons. And this turns out to be the pulse width duty cycle modulation control signal that's sent to the drive motors for these helicopters that we're using as models. And we can just do a frequency to duty cycle converter where we get a different duty cycle depending on the frequency at which the spikes are firing. And with the two rotors, if the two neurons are firing exactly the same frequency, you fly straight. If the two neurons are firing at slightly different frequencies, you yaw to either the left or the right. And this turns out to be really smooth graded control. This was the easiest part of this whole thing. Okay, now how do we wire all this stuff together to get a simple circuit that's going to mediate this search behavior? So we start off with some commands for clockwise rotation and counterclockwise rotation. And we have a split actuator for the bee right now that's under development that will allow us to control the amplitude and roll, pitch, and yaw differentially on the two sides for the control of the wing steering. And then we map onto this our optical flow neurons. We map onto that our heading interneurons for heading deviation. And then finally, the optical flow neurons project to odometer neurons. So this is our basic circuit for flight control for the search portion of the mission. Now, in the hardware, we use the Amtel AVR32 board, which is what comes stock with these computers. And we use the IC2 bus to map in a compass and the flow detectors. Then we have a releasing mechanism layer where we calculate from those inputs the neuronal activity. We have the neuronal network, and then this is a pulse with duty cycle modulation circuit that controls the actual rotors of the helicopter. So to mediate this behavior, we use a phenomenon we call reflex chaining. And probably the best example of this is the leech feeding system, where each phase of feeding in the leech causes sensory feedback, which triggers the next phase of feeding, and you just switch through these different sequences. So the first one would be to yaw to heading, to translate. One is completed with feeding to reverse heading, and then to translate back to the hive. So this is kind of how we would do it. And this is a simulation in LabVIEW showing the heading deviation neurons, the optical flow neurons, the control motor neurons here, and then finally this is a leaky integrator for the odometry. So we turn this thing on, it orients to heading using the deviation neurons, then it will, it's doing a little oversample there, then it takes off in the flight, it starts to integrate on the leaky integrator, it fires a spike. At this point the thing would go into UV mediated pollination, and then it reverses and flies back. So it does a little overcompensation there, wiggles around. And this is the biggest problem we're having with this right now, is it keeps oscillating when it's trying to get its orientation right. So this is it in simulation, and then this is now an actual model where we're actually controlling translation not with the rotors but with a motorized sled, so we can control the optomotor input. But the compass is controlling the orientation, and then we're actually using a leaky integrator, and when it gets to its required position it flashes an LED which turns the sled back in the opposite direction. So this gives us a chance to really experiment with the optomotor part. So here it's going, the leaky integrator is integrating, it gets to its desired distance, and then it'll reverse heading, and then fly back. This problem, this missing around getting the heading right, is something that was really very painful, I have to tell you. Okay, now for this homing on the flower, one of the visual sensors that bees use for directional sense is ocelli, and Rob Wood has already built an ocellus, and what we plan to do is to set these up in a 3x3 array where they map onto pitch and yaw interneurons to allow this thing to home in on a target. Now at this point, I want to kind of switch gears, because all of these things I've been talking about, homing on flowers, certainly stigmergy, are mediated primarily by olfaction. And olfaction has really been a problem for robots. If you know much about it, the current director of DARPA used to run the dog's nose program, which never worked out. And so people are really trying to find ways to get olfaction to work. And I think the key to this is a new form of molecular biology called synthetic biology. And what this does is to try to put an engineering framework onto molecular biology, where we treat genes as individual components, as the physical layer. We treat biochemical reactions as gates. We treat the biochemical pathways as modules, and then we treat the cells as computers, and they're connected in networks which would be analogous to cells and tissues. So we're trying to apply that framework to molecular biology. Now, how I got in this was a very strange phenomenon where the EPSRC and the National Science Foundation had a thing they call a sandpit. And what they did is they invited 30 of us to a resort in Virginia and put us up for a week. And we were supposed to identify the grand challenges in synthetic biology and form teams. So this is the team I put together. And our grand challenge was to deal with the I/O problem. How do you get sensors to talk to microelectronics? and how do you couple power with actuation. So we decided to build what we call a biohybrid robot, and this is it. It's going to be about 1 centimeter long, and it's going to be sort of modeled on a hatchet fish, and it's going to have some axial undulators, some sensor cells, a microelectronic brain, and then be powered by a thin film fuel cell. I mean, a thin film battery. We're going to try to use microbial fuel cells, but those are vacuous, believe me. So how does this work? How can we give a robot a sense of smell using living cells? So we start off with a chassis, and one example of a chassis would be yeast. And then we insert into the cells using a lentivirus vector some genes. And the genes we would like to insert is a receptor coupled to a G protein with a calcium channel, and then a reporter, which is aquarin and solinzerine. So the function of these genes is to respond to something that the Navy is interested in. This G protein couples the receptor to a channel. The channel allows entry of calcium, and then the aquarin reports by generating a photon of light when calcium enters the cell. Okay, well, it turns out that there is something called the Registry of Biological Parts over at MIT, and if you go to their website, they have a catalog where they list various genes, reporters, and then you can pick one out and you get a spec sheet like you would for any electronics part you buy. But what's interesting here is that there's a little button on the website, and if you're registered for the website, and they're very concerned about the do-it-at-home community, so it's pretty hard to get on this directory. But what they send you is this spec sheet physically. It's an 8.5 by 11 piece of paper, and along the bottom there's a series of red dots, and if you take those red dots and elute them in buffer, that's where the gene is. So you can just go out and get the parts, okay? So then you can couple those with a reporter, and you have what we call a device, which in this case would be a chemoreceptor. You can also take, for example, an opsin and couple it with a G protein to nitric oxide synthase and make a photoreceptor. So then we get from what's called the part level to the device level, and the level we're operating at is the system level. So my colleague Chris Voigt, who just moved to MIT from UCSF, has been working on some luciferases that give us light reporters to particular chemicals. This one is for arabinose, and he has different luciferases which respond over different wavelengths. And then my colleague Vlad Parpura is doing what right now is the most productive part of this program, which is to get muscle that we can control with light. And it turns out that muscles live as myoblasts, and you can make a stable line of myoblasts, and then with a compound called MyoD, which binds, it's a transcription factor that binds to this cassette here, you can transform the muscles, the myoblasts, into myotubes, which are the actual contractile elements. So what he does is he puts a TET-off motif here in this DNA construct, and that enables us to turn these genes off for MyoD with a compound called DOX. And then if we remove the DOX, the MyoD will be expressed and the myoblasts will form muscle cells. So we don't want it to form a muscle until we've actually built a device. And we want to keep the cells in the state of being myoblasts. So the way this stable line protocol works is you incorporate these genes, use an antibiotic to kill the ones that don't have the right genes in them, and then you take off the DOX and you form muscle. Now, to mediate excitation contraction coupling, we're also putting a gene into the muscle cells called channel rhodopsin. And channel rhodopsin is a G protein coupled receptor, which when it receives blue light, it allows a calcium current. And we not only have this incorporated in the cell membrane, we have it incorporated in the sarcoplasmic reticulum. Okay? So that means that we can shine blue light onto the muscle, And we can do this by taking the action potentials from our electronic neurons, having them gate an organic LED that produces the blue light, that will release the calcium, which will cause the excitation-contraction coupling to occur. So this is, again, a synthetic way of getting excitation-contraction coupling. Now, the beauty of this is this eliminates a whole bunch of cellular machinery. We don't need action potentials. We don't need a transverse tubular system. We don't need synaptic receptors. And so we can do a process called refractoring, where we actually take out a bunch of genes we don't need and make a really simple muscle, and we can hold it in the state of a fibroblast. And then the fibroblast can maintain dormancy. You can keep it for long periods of time. You can put the transcription factors on it, remove the docks, and light it up, and you've got a living muscle. And I really think this is really one of the most fruitful approaches to making artificial muscle. Now, just recently, there's been discovered a new channel rhodopsin that produces 4 times the current of wild-type channel rhodopsin, and it's almost pure calcium. And we've already got that gene in our muscle, so we're ready to go with that.
Joe, how are you going to say more about the muscle and how big you can make it?
We're just getting it to work right now. Give us a while on that one. That's a really interesting question. But, you know, I'm more interested in meat in a dish, you know. And I have a side story about this I'll give you. There's some interesting things we can do here. Synthetic biology is really, really powerful. So we need to build an electronic nervous system for this, and this will consist of a 4-cell oscillator that alternates on the 2 sides and then has a front to rear delay that will cause propagating undulatory waves in the body. And then the sensory neurons will feed into this central pattern generator to turn it on. The chemoreceptors, so these are our swim commands, and then these are the chemoreceptors. We really don't need this layer. We can have these impinged directly onto the neurons. So our minimal circuit is 2 neurons that would be stuck on either side of this robot. And I say this because building these circuits requires a fab, which costs, typically if you do a big fab, it's $150,000 a round. And we have a process in Massachusetts called MOSIS, where they do it for free if the circuit is small enough. And this is the biggest thing that we can build using the free MOSIS process. So that's where this is all about. Okay, so another problem is getting neurons to operate. Remember I talked about the Heinrich-Rose neurons, those are big boards. We have a colleague named Young-Bin Kim, and Young-Bin Kim has figured out how to make these operate at 800 millivolts, which is what we can get out of these thin film batteries using a 65 nanometer CMOS process. So we've already got these designed, and they're in the process of fabbing these now in Korea. So we'll have at least brain circuits built. And we're toying with the idea of using what's called epitaxial liftoff, where you actually form the circuit on silicon on insulator with a sacrificial layer. You build the circuit, and then you lift the circuit off the silicon on insulator. So you have a circuit that's about 100 microns thick and very, very small and light. And then that we would position in a robot. So we also have synapses worked out in subthreshold analog VLSI, and we've got this thing in simulation generating the alternating pattern. Now, how do you assemble this mess at this scale? Now, the standard way of doing this sort of thing is what's called inkjet printing. And in inkjet printing, you have a piezoelectric actuator that forces ink out of a target, and that splatters on some substrate. The problem is because you're using pressure, there's a cone of deviation, and it's very hard to get fine detail with this. So there's a new process that came out of Andrew Aylwine and John Rogers' lab at University of Illinois, which is called electrodynamic printing, electrohydrodynamic, they call it EJet. And here you take a microelectrode that might have a tip diameter of about 1/2 micron. You sputter coat the electrode with gold, and then you put a very high voltage potential between the ground plane and that microelectrode, and it calves off little droplets that are on the order of nanometers, hundreds of nanometers. And they've been able, using EJet printing, to print features of protein that are about 5 microns in diameter. So this is a very, very high resolution way of doing printing, But because it uses a glass microelectrode, unlike inkjet printing, which uses plastic hardware, you can use any solvent. You can make inks out of nanoparticles of gold. You can make them out of neurotransmitters, out of various modulatory agents. You can make them out of protein, and you can even print cells. Okay? So this is an example of a trace that is quite large. It's 70 microns wide, but that trace, this shows its physical cross-section. That trace will carry 1 amp of current made out of nanoparticles of gold. So it's possible to produce some pretty fancy electronics, and then you can print adhesive molecules or growth factors onto substrates and get cells to grow at very fine detail. So we can actually begin now to assemble living cells with electronics using this process of inkjet printing.
So that's what you mean when you say print cells?
Yeah. Yeah. Or you can just print an adhesive on the substrate and dip the substrate into a suspension of cells, and they'll glue themselves. Okay? So how would we make an undulator at this scale using this process? So I don't know if you remember the Mars spacecraft, the United States. They always had this gold foil on them, and that was a radiation shield, which you can buy by the yard now, and it's gold-plated Kapton, and Kapton is the plastic that your memory chips come in when you buy a memory board for your computer. That's made out of Kapton. So what you do is you take Kapton, and with inkjet printing, you print a resist on it. And then you put the Kapton, gold-plated Kapton in a solution of ferrocyanide and sodium hydroxide, and you etch away the gold, and then you etch away this photoresist with a solvent. And you're left with anodes and a ground conductor. Okay? Then you print on it the conductive polymer layer of an organic LED, and then the fluorescent emissive polymer layer, and then with some metallic ink, you print on a cathode. You coat that with spin coating with a dielectric layer, and then spin coat it with some kind of muscle repellent. And then you print a muscle adhesive. It could be polylysine. There's a variety of things. And incubate this with myoblasts. And then you wash off the docks, and suddenly you've got a tail with nerves, muscles, and an excitation-contraction coupling system. Okay? So that's one way that we can begin to build, using synthetic biology, fancy actuators. Now, photosensors, there's a very interesting phenomenon called photo-induced charge transfer, where you have a mixture of these two compounds, and when you shine light on them, they generate a current, so you can then form a photodiode on glass, and you can put filters on it for red, green, or blue, and then you can put a bacterial glue on that, or a yeast glue, and you can put the sensors right on the sensor cells right on the detector electronics and build a sensor system. So how would we integrate this mess? So this is the whole thing we want to put together here. So we would start off with a Kapton chassis. We'd put the actuator system on that. We'd glue the microelectronics on that. We'd put the electrode sensors on it, a thin film battery, and then incubate this thing with cells, and we've got a biomimetic robot. And this is what we expect to be doing in the coming year. Okay, I just recently got a ONR Muri with a pretty stellar collection. George Church just got elected to the United States National Academy of Sciences. So these people are really the cream of the crop of synthetic biology. And Vijay Kumar and myself are the two roboticists in this team. And the goal of this thing is to really enhance the variety of sensors that we have for applying synthetic biology to robots. So the whole idea here is to engineer biomolecular sensors using synthetic networks. These will then be used to create what we call microbio robots. And these microbio robots will form the sensors and in some cases actuators, and then they will communicate with a chaperone robot, which will be both the lobster and the lamprey robot. Okay? So this is the big picture. And this grant just started a month ago, so I'm kind of giving you an idea of what the future is here. And so what they're fooling around is with programmable bacteria. And one of the things they've been able to do is to take a lot of receptors and modify them very specifically for particular compounds. And they have a two-stage sensor which first hydrolyzes high nitrate compounds and then will respond basically to things like RDX. They're also working on a structure within the bacteria called a stressosome, which is sort of the final common pathway by which bacteria deal with environmental stress, temperature, hydration, whatever, and getting that to operate with millisecond responses. And also they're working on genetic memory, such that if these bacteria were to contact RDX, they'd remember that they'd done that, and you could interrogate them at some later point and find out. So they've got a machine, I mean, this is just completely crazy, where they design what genes they want. They go through the library of biological parts. They assemble using something called this is a CAD environment for defining gene networks. They actually build the device that they want, and then they've got wet robotics that will synthesize it. And this is the part that is, this is why they call it interdisciplinary research. You know.
Just wanted to check how much material you've got left.
About 2 more slides.
2 more slides.
Yeah, I'm getting really, really close to the end here. I mean, actually, I may have 4 slides. I'm not sure. Okay. So then Chris Voigt and I are working on some fancier new designed photoreceptors that we're going to use for cellular electronic communication. And then the group at BU has this thing where they put dextran and then this stuff called SU8, and then they attach bacteria to the SU8, sacrifice the dextran, and they have these little things we call microhybrid robots, which are little flagellated bacteria swimming around on their own. And Pam Silver is actually working on inserting photosynthetic genes into these so they'll be self-powering. And then they've got a fancy command and control system for controlling these, which we're going to attach on certainly the lobster robot for using these for various kinds of sensing operations. Now, the last thing I want to finish with is kind of one of my pet projects that really kind of takes biomimetic robots to the limit, I think. It turns out that in the United States, the second largest contributor to the balance of trade, second only to oil, is imported fish. We have basically fished out everything. It's a huge economic issue. And the solution to this is fish farming. Now, most fish farming goes on, about 90% of farmed fish is produced in India and China under conditions that are health hazards. I mean, you don't want to be eating this stuff. There's toxic pollutants. It's just really bad news. They dose these things up with levels of antibiotic that are unheard of in the livestock industry. So it's really quite a nightmare. So fish farms, this is an existing fish farm up in Lubeck, Maine. And the biggest problem with fish farms is that nobody wants these in front of their waterfront property. It's as simple as that. It's just completely crazy. Secondly, they're navigational hazards. I mean, imagine running into that at night in your boat at 30 knots, you know. Third, they get broken up by storms, and they lose the fish, they lose the whole thing, so they're a really capricious fiscal investment. And fourth, they're very highly polluting. So these fish are putting out a lot of waste, they really change the environment. And fifth, working on one of these devices is really hazardous. And so there's all these OSHA standards that go in, the labor costs are over the top. So for this reason, fish farming just doesn't occur in North America. There's the biggest example of fish farming is people out in Iowa and places like that growing shrimp in ponds or catfish. But there's nothing else really going on. So our idea is to build a biomimetic robotic fish farm. And the idea is that this would be a tank that's about 60 feet in diameter and about 15 feet deep and would be anchored to the bottom. And my colleague April Gu is a chemical engineer that's come up with a 3-stage biofilter system that takes out nitrates, phosphates, and solid waste, and she's working on ammonia right now. And our idea is that this thing looks like a big sea anemone. So we would just control this thing with an electronic nervous system that's based on seleniterate nervous systems, which is another project I have going on in my lab that's supported by Schlumberger. And so the whole idea then is that you take this thing and pull it underwater. And if you pull it underwater, you can use wave energy to power it, so it becomes a green technology. People no longer have this in the view of their waterfront property. If it's far enough underwater, it's no longer a navigational hazard. And if you get into a situation where there are major storms, you just pull it farther underwater. It turns out wave energy falls off with a third exponential of depth. If you get this thing 50 feet underwater, you can ride out sea state 8. So it's a really feasible way of doing it. But then you got to control this thing. And so I have a colleague, Dave Brady, who's a wireless expert. And the idea is that we would use sonar communications up to the surface and then Wi-Fi this to the shore, so you could actually control this thing with your iPhone.
And pick the fish you want.
Yeah. So you could have a video camera and you could be watching what's going on in the tank. And so it really solves the whole labor maintenance problem. And I don't have it here, but we have a feeding mechanism which you could change out about once every 2 weeks. And it turns out this is the most amazing thing, is in that first fish farm I showed you grows salmon. It's up in Lubeck, Maine. And it takes them 16 months to get a salmon from smolt up to adult. That's about an 8 to 10 pound salmon. And they feed them herring meal. And the herring meal is basically using fish to grow fish. That's one problem. But the conversion ratio to get to 16 months is 1.3 pounds of herring meal per pound of dressed salmon. So these things are almost at 0 degrees, so they have very low metabolism. So you get this incredible conversion ratio. So economically, this is a huge profit machine. And one of the ideas is to actually, instead of feeding fish, they normally would be eating arthropods. So there's some guys in the United States that are working on taking restaurant waste and using that to grow insects, and then taking the insects and processing them into food. So then you solve, this is an even more green technology because you get rid of all this waste. So it's really an idea where we could use biomimetic technologies to solve what is really a societal problem. And I think that's the thing that really...
Can you have some biomechanical mosquito catching robots that will bring mosquitoes to
Believe
your
me,
fish farm?
no. I have a story from Lanzarote, and Frank lived this too. Mosquitoes are the worst. You know? No, I got nothing good to say about them. So we call this whole endeavor neurotechnology, and we argue that it is to neuroscience what biotechnology is to molecular biology.
Very good.
Well, thank you very much. I know everyone's getting hungry. The traditional Barcelona is to have a late lunch, so but this is late even by our standards. But I think we can have a couple of questions since it was such a fascinating talk. Yeah. Well, I think it's one of the back first.
Oh, students first.
Well, Andy's not really a student.
Well, go ahead.
Go ahead.
No, I was fascinated by the fish thing. I think it's interesting. I mean, pigs were used for that very reason. Pigs in the 16th century were there to eat the rubbish, and it's still the greenest meat to eat if it was, if they were brought up on waste. It's a way of transforming food waste, into food. But my question's not about that. With the robot bees, have you got any plans to use more vision on them? Because, I mean, bees navigate visually.
Yeah. Well, we have these optical flow chips and the ocelli. Those are the two things that we plan to use.
I mean, I'm sort of reconcile it rather than flow waste.
Yeah, there is part of the group, Todd Zickler, that's actually working on imaging and using landmarks, and he's got some schemes for doing that with some of Jeff's vision trips. The sad truth of this project is that the bee is going to weigh 500 milligrams, And we have been allocated 55 milligrams for the brain and the sensors. Okay, the little brain I showed you is about 35 milligrams. Okay, so we've got about 20 milligrams for the sensors. So we're probably going to go to e-jet printing to build those sensors for that purposes. Yeah.
And what's going to be the energy that it's going to be needed for each
Excuse me?
Energy, we're going through requirements.
For the bee? You really, you know how to probe the soft underbelly. Right now, with the battery, the bee is good for about 3 minutes. With a solid oxide fuel cell, it can do probably an hour and 15 minutes. But the solid oxide fuel cell operates at 350 degrees centigrade. Nobody wants us to land in their lap. You know. This is the most active area of this project. I have to tell you. I mean, we just had our 2-year NSF review last Thursday. And I would say 70% of the questions we got from the reviewers were exactly on that subject. I mean, they think, if you saw how this manufacturing process works, I mean, they're going to be able to build tens of thousands of these. It's just outrageous. It just flabbergasted me. I had no idea that mechanical engineering was to this point. The power is really the weakest part. And the weight budget is nightmarish. I mean, if you saw the pie charts for where the weight distribution is going, so they equate power to weight, and generating the power. The actuators use 95% of the power of this thing. So it's just, you know, like one thing I learned in dealing with engineers is that everything is a compromise. Yeah.
Biomimetics and power harvesting. Yeah.
Well, I mean, this idea with cyberplasm is to use real muscle and couple the energy generation of mitochondria with the production of force, and that's really the obvious way to go. And you know, I really think the question you were asking about the scalability of the muscle, if we can get this to scale and we can build big muscles, but you know, there's this whole business going on. I don't know if you've seen recently, just 2 weeks ago, there was 3 articles in Nature where they discovered transforming factors that will convert human fibroblasts into neurons. Okay? And they can produce dopaminergic neurons. They can do this both with transcription factors and they can do it with microRNAs. Okay? There is another paper by some Japanese workers where they took human stem cells and formed an eye cup that had a reflecting layer, a retina, and a lens out of stem cells. There's another paper where they generated a complete vasculature system with a beating heart out of stem cells. And most recently, there's one where they generated an anal sphincter out of stem cells. And when you think about this, they're doing this from fibroblasts. So this becomes personalized medicine. And I actually have a proposal under review right now to build a pacemaker using human fibroblasts, which would be a personalized pacemaker that would operate on glucose. So I think the potential for actually engineering medical devices using human cells through synthetic biology is the future of medical devices. And once you get away from batteries, then you're home free. Yeah.
Okay. Well, I think we a couple more questions. So they have to be easy questions. Let me try you, and then Paul, and then we'll take Paul.
Easy question. When in the part of your talk where you were describing the robots that went underwater, and you said that you had this robot down to about the cost of its parts, I missed whether that was the robo-lobster, the robo-anemone, or the robo-lamprey, and I missed where the sensors and actuators on that robot.
Okay. Well, that's a simple question. Well, pretty much that's a generalization of the manufacturing process. Is that once you start mass manufacturing, as soon as the part count goes up, I mean, very quickly, if the number of units that you're producing the cost of the parts become the governing cost.
What are your sensors? What were the sensors?
Oh, on that robot, on the lobster and the lamprey robot, we have a compass, we have an accelerometer, we have flow sensors using the antennae, and we have bump sensors. And they're all made with MEMS parts that are $2 parts.
Oh.
Yeah, so if you talk about the behavioral control, right, and some of your point is very much to say, look, we can do this in a very minimal fashion. But you can do that when you live in a minimal world.
Yeah.
And in some sense, the applications you're describing, these are not necessarily minimal worlds. So there will be many problems there of, let's say, stability, local minima, behavioral space, etc. So how will it actually generalize the real world environment?
Okay, well, our approach, and that's a really well taken point, our approach is to really integrate biology with robotics. So in my lab, I have one guy that's building lamprey robots. I have another guy that's recording from central interneurons in freely behaving lamprey. And we have a way we can record from all the giant neurons in the reticulospinal system. I also have a guy building a lobster robot, and I have another guy that's doing the analogous experiment in lobsters. So our approach is that when we find a point where the lobster fails, or the lamprey fails, we then take the real animal and put it in exactly the same situation, figure out how it works it out, hopefully record from the neurons while it's doing that, and engineer that back into the robot.
But that's a methodology we all agree to,
Yeah.
right? But the point is, the solutions you showed us now are not very scalable, right? These minimal solutions, if you would put them out in the ocean, for instance, they would get stuck very quickly. There's a few obstacles around, or there's some current around, and so on. Or your little bee robot is going to break. With, let's say, in a little bit more complex world, it will get lost.
Okay,
right? Very difficult.
well, let me get back to the chaos.
Yeah.
The whole idea of the chaos, and the hardest part for us was figuring out how you get stuck. How do you determine when you're stuck with the level of sensors we have? And our approach is to do this accelerometry thing, to try to get something, look at the acceleration profile. If we get the wrong acceleration profile, we try something else. If that doesn't work, then we just start dialing up the chaos and explore the full parameter space until it gets out of it.
But that's not necessarily what your lamprey is doing, right? Or what your insect is doing.
That's what we're working on right now. That's in fact, that Anthony Westfall is working on that, is now trying varying chaos at different points within the network to see where it really makes a difference.
It was just, if you look at, let's say, a bee brain, where you go from the antenna lobe for the chemical sensing system to the mushroom bodies, the neural cerebrum, and so on.
Yeah.
These are pretty structured systems.
Sure.
We have no clue what they do.
Exactly.
But it doesn't look like they're producing chaos.
It depends on when the conditions under which you're recording from. Now, where I have the most experience in this is recording from the circumesophageal connectives of lobsters. And when you watch at the patterns of individual neurons, when they get in a struggle-like situation, which they do periodically, it looks like chaos to me. I mean,
this was not
I haven't calculated the Lipinov exponent.
Right.
I think we have to stop there. Thanks again.
Okay.